Jurassic World.
Good morning, Richard.
Oh no, not a problem. I had fun. I was being
cool.
a comedian. I was throwing in some
That's
jokes.
awesome.
You get? I'm sorry.
I was throwing in some jokes. We're getting some good.
Oh, good. A little
Laughs.
soft shoe, a little dance,
Yep, yep.
song and dance.
They shot a bunch of Transformers in it. Oh shit.
If I do it like
You should
on
watch.
the channel.
You should watch Shameless. It's
Series.
the most, but this is like you can't turn away. It's like a perfect train wreck that you can't turn away from.
It makes your life look so much
It does. If you ever feel bad about yourself, watch Shameless.
Because
Shameless is that after a while, you want to root for these people.
And then they let you down.
For you that
If they were your,
They do.
well, if they were your own family, you wouldn't accept it, but it's funny to watch.
I know, it is. I love that show. It's a great show.
It is. I know. It's starting up again in September, I think.
You know what else? I noticed the biking show.
Game of Thrones. That one.
Oh, Danielle loves that one.
I only watched a few episodes. They cut off a horse's head and now it's done.
I'm like, no,
Yeah. And that's it.
So, Nader, do we need to wait for anyone, or should we
You
just...
can go ahead and start. People can start trickling in. I know some people came in and then walked out because.
Oh, because I wasn't here.
So yeah, you can go ahead and
No,
start.
I'm the oldest of 7. I'm used to having people leave when I walk in the room.
Ah,
Hey,
That's just not.
I wasn't a bully, honest. Well, glad to see you all here. I thought it'd be kind of interesting to talk about something other than a neuroscience-related topic because, for one thing, the immune system is something that is taking care of us. All of us who have healthy immune systems, at least, taking care of us 24/7. And I think you could probably say the same for every other functional system in the body. But the immune system does a whole lot of work, and we don't really talk about it a whole lot. One of the things that has only become evident within the last couple of decades is the significance of different kinds of blood cells in terms of their ability to fight infection and cancer and other situations that you might say are things that are foreign to our body. One of the things that's really a signal property of the immune system is that when the immune cells themselves develop in the body, they're basically trained, if you will, biochemically to not recognize our cells as foreign. If they did recognize our cells as foreign, there would be a lot of trouble. They'd be mounting an immune response, and that can blossom into all kinds of medical issues, and that's more or less in a very superficial way the basis of certain disease conditions where people have developed immunity against their body. Not a good thing to have. Anyway, there's a group of cells that are called resident memory T cells that were discovered not that long ago. These are cells that, as their name suggests, they are memory cells. And memory in a cellular basis basically is biochemical. It means that in the case of these cells, there are proteins that are on the surface of the cell. As I said, they're expressed on the surface of the cell. It's something these cells learn how to do in their situation, and because they are primed to be able to do so, it's part of their lineage. And lineage in the cellular basis is probably even more critical than in a personal basis, because lineage means that these cells, if they survive, are going to become something along a sequence of capabilities that are usually described in biochemical terms, like in the genes that are expressed, in the proteins that they either secrete or put on their surface, or proteins that are generated to help to change the inner biochemistry of the cell, capabilities of the cell. So we start off with the immune system, and probably everybody's seen something kind of like this. As we all know, there's basically two large chunks to the immune system. One of them is the antibody-based thing. You know, if you've ever done antibodies with animals or something like that, you immunize the antibody or immunize the animal by injecting something into blood or into muscle. Wait a while, do it again after that, and after the second time, you've got huge quantities of IgG floating around in the animal's blood. There's a whole lot of stuff that underlies all of that. But basically, what happens is you're stimulating B cells to secrete these proteins. The other half of what's going on in the immune system is a cell-mediated response, and you know that's what happens when you have infected cells that are activated, and it stimulates a sequence of different cellular activities. Cytotoxic T cells can, because they will have a receptor, in quotes, which is an antibody-like protein on their surface that recognizes something that's foreign. Cytotoxic T cell will snuggle up to that. There'll be a binding event with those things, and then they secrete something that will kill the other cell, which is why they're called cytotoxic. One of the interesting things about the immune system is it doesn't just happen and go away. It's retained over years. So, for example, if you were to, in the laboratory, to immunize a mouse or a rabbit, take out some blood, you go through the procedure, you have to immunize them a couple of times. After 6 to 8 weeks, you've got blood that's very high titer, that is a very high concentration of antibody to what you have immunized the animal against. If you come back and bleed that animal again a year later, 10 years later, the titer may have decreased, but it's still there. So there's immune memory throughout that system. It happens in the humoral response and also in the cell-based response. In the cell-based response, the thing that pretty much keeps everything in order are these so-called memory T cells. And that's pretty much the center of this little talk. But what part of what's interesting is about this is that these two systems work together. So T cells, they arise in bone marrow. They migrate to the thymus gland to mature. They require that the antigen be bound to cell membrane-based molecules. This is MHC is a major histocompatibility complex. It's one of the things that you can distinguish people by differently. If you just take a blood sample, people have different MHC proteins depending on their lineage and whatever. But different MHC molecules, different classes of them have different activities that they will do. So I mentioned that we have helper T cells that are involved in the immune response, cytotoxic T cells that will recognize a foreign invader cell like a bacterium and can snuggle up against it and kill it. And suppressor T cells can kind of turn down the whole system. I don't know if you can read this, but a killer T cell is the guy with the gun, and he's coming up against virus-infected cells, and he can terminate them. So he's the Schwarzenegger of the crowd. The critical steps involved in T cells becoming something like this is that a relatively freshly made, naive T cell, and these cells turn over, more or less continuously in the body. I don't know what their lifetime is in the body, but it's probably more than a day. But certainly everything turns over in your body, so you like to see it refreshed by a new population as time goes on. So a naive T cell, once it hits an antigen that has the MHC protein and the antigen next to it, and that causes the cell to proliferate. So the cell begins to mature, and it starts to express different proteins on its surface that recognize that antigen, and it divides and divides and divides until you have 2 to the n, a much larger number of these cells. And they can also differentiate into memory T cells that have the capacity to recognize these things, and there's other effector T cells. So, much like gall, for all of those who've had Latin, T cells may be divided into 3 parts. I don't see any Latin lovers in the room. One of these is to circulate between blood and secondary lymphoid organs. Another is to circulate between blood and non-lymphoid tissue, and the third are cells that do not recirculate, but they are tissue resident, and those are the focus, the ones that actually live within other tissues. And they're not there by mistake. They have specific function that we'll be talking about, and functions and structures, of course, go together in biology and everything else. So how do these memory T cells live? What's the lifestyle of your basic modern sophisticated memory T cell? They first migrate into non-lymphoid tissue. This was first discovered by somebody who was a graduate student at University of Connecticut. He discovered the notion that these cells migrate to non-lymphoid tissue. And they live there for a relatively long time, and they're functionally distinct from other T cells. Come on. There we go. So, as everybody, you meet somebody new, you want to know what their house is like. What's their house? Well, memory T cells, they're living in tissue. They're not incapable of moving around. They're not incapable of responding to their environment. But they do have particular biochemical means of recognizing where they're supposed to live. Basically, they have a key to the front door. So they express proteins that position them at a site of infection, which is a great place to have T cells because infections don't just go away, as anybody who's gotten a little pus or whatever, and that whole business is going to be around on your arm for a few weeks as it resolves. Part of the resolution is due to these memory T cells that are just hanging around there and some other effector cells. So they have particular proteins on the surface of the cell that allow them to recognize a location. Let's see, they also can identify a particular kind of collagen that's involved in the basement membrane that is the structure that cells sit on. Cells don't just hang out there doing nothing, they're sitting onto something, at least ones that are attached like skin cells, internal organs, that kind of thing. They have proteins that help to maintain their shape and the integrity of the cell so they can function, and they can block gradients of something. I'm not exactly sure what this is, but evidently this is some nasty thing, S1P, that I didn't have time to look into. But they're capable of doing all of those things, and the point of all of that is that they're going to be sitting in the tissue where they are now primed to respond to a potential quote, insult, that is some biochemical thing that is foreign to your body, and they've already been primed to respond to that. So this is what happens after they've been primed. So dynamically, what happens when the memory is turned on, you start off with a naive T cell. And naive means they have not been informed by their environment to become something beyond what they were. So they go from the naive T cell to a stem cell that is a memory stem cell. And that means that they can still differentiate a little bit more, but they're down the path towards memory. And then the next step of differentiation is to go into central memory, and the third is effector memory. Effector memory means now that once they see that unique thing that was priming them way back here, they can respond to it. And then finally, they become terminal memory, effector memory cells. And the last ones in line are the TRM, which is resident memory cells. The difference of all of this is once you get down to resident memory cells, they live in that tissue. So it could be any of the organs of your body. They just live there. They've been primed to see something back here. Maybe you got a splinter in your finger. You see pus after a couple of days. Oh, no big deal. It's going to go away in a week. Part of it doesn't ever go away. Part of it sticks around for years. And that is largely mediated by things like these resident memory T cells in the offended tissue. The whole differentiation pathway, each of these steps, they're relatively discrete, but within a population there's a lot of overlap, but they're relatively discrete. And there's a sequence of these things that are due to the exposure to the antigen, that is the protein thing that caused them to respond to their environment changing, and to inflammatory cytokines that are released by other cells. So there's a whole sequence from the most naive cell all the way through to points where you have a resident population of these resident memory T cells that hang around for as long as years. And the T cells actually do kind of move through the environment. They have migration patterns. Generally speaking, there are other classes of T cells besides these. There are some that live in your bloodstream, and they're involved in various factors in the immune system. You know, if you have a bacterium that comes in, you have particular ones that respond to that. If you have something that is a soluble antigen, like a protein that's secreted by a wasp that sticks a stinger in your arm, and the T cells come in there, and some of them are going to be coming up against that little thing and where the boo-boo is and taking care of it. So there are migration patterns that you can see within different tissues. Part of the backstory to the whole discovery of the fact that we have these things is that this guy Manopust at Connecticut discovered that there were these unique cells that could only be found in certain tissues that had been primed to something. It was later that they found out that these unique cells would also identify that what they were primed to. So it was a really nice chain of discovery. The guy, that was less than 20 years ago. The guy's got like 200 publications out of this thing. So it's worked out for him. Anyway, so you have a class of cells that basically go everywhere in the body. They travel through the blood. They go into lymph. They go into whatever. You have a class of cells that is much more tightly defined in where they go. So there are several classes of cells that will travel through different compartments, if you will, of the body. And then you have the resident memory T cells that stay within the tissue they live in. Hello. So, if you get a vaccination, the response to the vaccination includes these guys. You're starting off with these naive T cells. You have a precursor population of cells that will respond to whatever, just because your immune system has kind of divided up responsibilities into the future potential of having something come into the body that the body doesn't like. So these precursor population will then proliferate. When you start off, you don't have any resident memory T cells that recognize the thing because that whole sequence hasn't begun before the vaccination, smallpox, whatever it happens to be. Soon after the vaccination, you have this small precursor population has started to proliferate into different groups that can be separated functionally and in other ways. And there's really nothing in resident memory, even late post-vaccination. So it takes a while before that entire process is continued. And this thing is, sorry, it's a little blurry, but anyway. So let me see if I can start off from the beginning. If we start in circulation, on this side. If you start in circulation, within epithelial tissues like the skin, you start in circulation where there's a boundary between the two. You have something that is foreign antigen, activates the cells. The activated cells go into circulation, and you wind up in circulating memory T cells, which are not the resident memory T cells. Resident cells have a different function. They not only live in a different place, that is, they don't circulate, they stay in the tissue, but they have a different function. So you have circulating memory T cells, but you also have this sort of thing that's shunted into epithelial infiltration and becomes part of a cycle of reasserting the memory T cells in their function. So when a pathogen comes in, anything, these things, and it stimulates cells and they secrete all kinds of effector proteins, you get recruitment of naive cells into a population, they begin to divide, and you get activation, and what then happens is that you get the recirculating memory T cells, and you wind up with cells that only remain in that tissue that are the tissue-specific ones. And what is it they're there for? At the high level, they're there to maintain tissue homeostasis. That's really what many of the organs of the body are there for at some level in what we do. They're there to make sure that we are capable of doing what needs to be done throughout the day. So keeping antigens, foreign invaders at bay is part of what resident memory cells do, and they do it in the tissue that's been injured. So they're primed for that fact. So if you get a wound and you get a lot of exposure, these memory T cells, which are the green ones, will will get triggered a various number of protein factors, interferons and tissue natural, TNF-alpha and things like that, become effector proteins that cause the protection and regeneration functions of these cells as they live in the tissue. So what does this mean? This means that we have these cells that live in our tissues. They're central to the immunity and homeostasis that is keeping everything going the way it was going. They represent a long-lived and tissue-embedded immune memory that wasn't before even considered. Everybody thought, well, immunology is all in the bloodstream. But it takes discoveries like this to really figure out all of the details of this. The tissue-embedded memory is related to local challenges, and it is triggered by a history of infection and inflammation. And its ability to retain locally underscores its core function. It's also critical to cancer surveillance. Obviously, cancer can be triggered to start in virtually any tissue in the body. So having cells that say, I know what normal is a good thing. Having cells that are triggered to tissue transformation, otherwise known as cancer, can be a good thing as well, as long as they can control cancer. So these cells express these checkpoint inhibitory receptors that will kill cancer. The resident memory cells also trigger some adverse effects, as most things do, related to tissue inflammation. And it is possible to generate pathogen-specific memory cells in any location. Thank you all for coming.